First, we have to consider existing tools that may be used. Uh, the first of which we're going to consider is very highly used for confocal microscopy at the Institute. It's called IMARS, and that does a range of different microscopy approaches, including wide field, confocal, light sheet, two-photon, EM, SLEM, OPT, and so on. And there's more information available here, and there's the example paper where you can see an example of how it kind of works. An alternative that is less commonly used at the Institute is for looking at kind of 3D reconstruction and tomographic reconstruction of cell structures and cell movements. And there's more information at this site here. There's also a tool called ImageJ, which is a general image processing tool. It's multifunctional. It is wide use at the Institute. And you can see here for more information. There's more information at this PDF link as well that covers all the basics for using ImageJ. So what are we trying to do with bioimage analysis? What we're trying to do is extract information from data, image data in this case. So we're taking our image, we're going to record it using a suitable tool, process it in some sort of way, analyze it, perhaps do comparisons. That will generate our results and give us some sort of scientific understanding. So that's our general flow. We like to think it's linear, but when you think about the steps here, where we have a research question and sample preparation, then imaging, the processing, statistical analysis, linking to other results, and our understanding, a lot of this requires back and forth. I may have to refine a research question if we find there's nuances in sample prep, or maybe the analysis generates other factors that we need to control for in our initial experiment. So this is not a linear process by any means. It will jump between these different steps, just like any normal scientific endeavor. What you might want to achieve in any analysis is things like, such as comparing control cells versus exposed ones, looking at movement over a series of images, maybe using different channels too. And we want to look at the numbers of different virus particles, their sizes, and things like that. So we can think about what we're doing in this kind of work. So in essence, what we're trying to look at is things like deconvolution. We're trying to improve image quality and use our knowledge of microscopy and the light that we're measuring to improve the images that we see, depending on what we're trying to identify. We also want to think about how this is essentially solving the inverse problem, where we infer our data from the factors that are producing them. So we're trying to kind of make that inference process. We also need to denoise or remove noise from the image using our understanding of noise statistics, bearing in mind that noise is a stochastic process, and therefore you can model it and integrate it into our different approaches. There's an example paper that we'll talk about next. So this example study is about virus factories in the cytoplasm of cells that are infected with some tagged coronaviruses. And what the paper is looking at is whether they merge or not over time. So coronaviruses are non-enveloped bisegmented dsRNA viruses with a dsRNA genome. They replicate in these inside virus factories in the cytoplasm, so the small compartments. To create them, they co-opt endosomal membrane components. These viruses are known to reassort, and that's a major concern because they make more virulent viruses, where it's unclear to what extent virus factories are involved in that process. So if we look at the structure of the virus particle here, we can see there's different proteins, VP1, 2, 3, at the RNA inside the two segments of it. And you see that there's different proteins inside as well. So the questions will be things like, where are the virus factories? How many of them are? Do they move? Do they interact? Do they merge? And to assess that, they examined 2 rescued infectious bursal disease virus cultures. They fused the marker to a VP1 polymerase and used DF1 cells for transfection, and then we're able to use some staining with this compound called bioidentical derivative of red fluorophore RECIPERIN, REACH for short. So see here, is these are the reporter strains. So they have DFP to create that nice green color, and then magenta with this TC tag as well. So you can see there you have to use the BP1, 2, 3, 4 as the backbone and created these two different tags so they can see which segment had be or present inside these various particles. So you can see initial images here. So they're able to take these DF1 cells, transfect them with the plasmid expressing GFP before transfection, and with the plasmid expressing GFP or infection with the virus as well. So you can see that on this side here, GFP on its own, we see nothing. GFP 11 on its own, we see nothing. Two of them together, we can see that we see high expression. And then we are able to visualize the virus particles here in green. So you can see that there's clearly evidence of abundant infection of these different cells. And you can see the virus factories there. To look at the DTC tag, again, the DF1 cells were tagged with the same virus stained with this bioidentical compound. And this was compared to the recombinant one. So this is the control. And again, we can see the virus factories there stained. So what they're able to show is that the green or red fluorescences in the cytoplasm were co-localized with the VP1 and dsRNA segments consistent with virus factories. And they found that virus factories were very dynamic structures coalescing in the cytoplasm. Small virus factories move faster than large ones, and the rate of movements and dynamics depended on the internal cell microtubule network and actin cytoskeleton. What they saw as well is that resortment was dependent on this virus factory coalescence. So that's key to this process of virus reassortment. You can see more of this in the following images. So you can see you have the DAPI staining, the wild type dsRNA staining, and then the merging of these different images here on our right-hand side. What we see in the second panel then is also a link with GFP as well as the dsRNA and DAPI staining and the merged ones. So you can see I've got two different cells and stainings there. There's the wild type again, and then the GFP again at the end. So what we're looking at is the GFP signal as a marker for these virus factories. So we can see them clearly there. A and C show the DF1 cells infected with the wild type PGB98 viruses. That's that one and that one. B and D, they're transacted with the plasmid expressing the GFP1 and then the other GFP infection with the virus itself. The A and B are cells with the cells are stained with DAPI or antisense dsRNA, whereas with the C and D ones are stained with anti-VP31 mouse antibodies. So here what we can see then is that these virus factories are clearly coalescing around the cell inside the cytoplasms. And we can then monitor their movements as we go along. So what they saw then is that with the look over time, so 10 hours, 12, 18 hours, 24 hours, is that these virus factories coalesced and moved over time. You see there that they're aggregating here as we go through time. They again performed similar experiments to achieve this this kind of observation of them over time. So you can see that again here where they're looking over time, 10, 14, 18, 24 hours. They're able to look at the cells on their own, GFP labeling, linking to VP1, and then the merged image altogether. So they're showing multiple channels here that they can merge together. So they found that discrete virus factories from different strains of IBV in the cytoplasm coalesce throughout infection, and they performed similar experiments here to provide evidence of that. Two metrics that we're going to use that they used. The first of which might be new to some of you, is called Manders overlap coefficient, MOC, and they're able to look at the overlap between the red and green foci in any particular image. So that's looking at the overage. Was implemented with image A in this paper, but it's possible to do with R. We'll look at how to do that. Just looking at the overlap, or sometimes referred to as co-localization, and it's looking at the pixels and to see above the background to what extent the green and red areas overlap. Its values is scaled between 1 and 0, and it's measuring the magenta. If I say red, I mean magenta and red-green channels. And they're able to see that the correlation seem to increase over time on average. The other one we'll look at, which is similar, is Pearson's correlation coefficient. PCC are just the correlation coefficient value. And then it, as you know, measures the covariation of the magenta and green channels over pixels. So that's what we'll look at later in this course.